Welcome to the high content screening system. It's covered underneath here. I'll in this short video introduce how to turn it on and the basic operations of this system. First of all, there are two buttons, one for the microscope, turning on all the hardware components. If I need heating and CO2 control, then I also turn that on. Heating and CO2 is controlled by these two boxes, and they are manually controlled with it's quite straightforward. Just follow the arrows and press on and off. Then I need to turn on the camera, which is turned on manually behind the screen, like this. And you see the color change from orange to green. You turn on the touchpad, so also giving number 4 here. And the last thing is the PC. So at this point the PC will recognize all the components and we're ready to start imaging. So now we're ready to insert a sample, and in this system you can put in all kinds of samples. So this stage insert is equipped for plates. So let's say we have a 96 well plate. I just move the spring and insert my plate here, and it's ready at this point. You may have slides or something else, then you can take it out simply by removing it. And here I insert a holder for slides. And I always use the extreme position, so you don't want to use the middle position, because that is hard to calibrate in the system. So if you keep it to the left, then it's easier for you to calibrate the system. So slide it in and you're ready. So now I put my sample in the right position, close and turn down the light. So now it's dark inside. I start scanner acquisition software, and you can simultaneously start scanner analysis software. Okay, so in this system the software takes control, so the touchpad, you will not use that. You will use XY drive over here and the set focus here, and you can use some of the functions for shutting the light, for instance. Everything else would be handled in the software. Okay, so we are done imaging and we want to shut down the system. So all the objective lenses are dry, so we don't have any oil or water to clean. I simply shut down the software. And turn off the PC. Then you turn off the touchpad and you are sure you want to exit. And just behind the touchpad you have the power, so press that. And you take out your sample and close it. And then you power off the camera. And these two buttons over here again for the heating and for the microscope and all the parts. So finally we cover the microscope. And you note the status and when you were here in the logbook.